What gene drive approach are we currently exploring to reduce the number of malaria mosquitoes in Africa? At Target Malaria, we aim to tackle malaria transmission at the source. To do this, we are working to develop a genetic modification that could reduce the number of malaria-transmitting mosquitoes in sub-Saharan Africa. The genetic modification we are currently investigating targets the fertility of female mosquitoes to make them sterile. We use gene drive technology to ensure that this modification is spread across the target mosquito population from one generation to the next. Let's take a closer look at how this approach works.
We start by inserting a genetic modification that disrupts a gene essential for female fertility into mosquito embryos. Mosquitoes have 2 copies of each gene, one on each chromosome, and chromosomes exist as pairs. During the production of eggs and sperm, the gene drive system recognizes and cuts the unmodified chromosomes, which then repair itself, copying the genetic modification from the modified chromosomes. This process, called homing, ensures that both copies of the chromosomes in the mosquito reproductive organs now carry the genetic modification, and that this is inherited by nearly all the progeny.
When mosquitoes with one copy of the gene drive met with wild mosquitoes, they will pass the gene drive to nearly all of their offspring. Each will carry one copy of the gene drive and will replicate the homing process generation after generation. Over time, the gene drive will pass to enough members of the population so that females and males who each carry a copy will mate. Only females that inherit the gene drive from both the parents will be sterile, while males can still reproduce and will keep passing the gene drive to future generations.
As more and more females become sterile, the population of malaria mosquitoes would decline, ultimately leading to less malaria transmission. Development of gene drive mosquitoes is still at the research phase, and all tests are done in contained laboratories. Releases in the field will happen only after receiving regulatory approval and community agreement. Our initial approach focuses on targeting 3 closely related mosquito species that are the main vectors of malaria in sub-Saharan Africa. Because the mosquitoes themselves do the work, this approach could provide a sustainable and cost-effective tool to complement existing interventions and reduce malaria transmission in this region. Together, we can end malaria.